Welcome back, students. Today we are going to discuss part 8 of unit 9, that is application of biology. Before I start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In today's video lecture, we are mainly focusing on antibody, vaccine, and antisera. So, what is antibody? Antibody molecule has a 4-peptide chain. This molecule is having 4-peptide chain. As you can see this is a Y-shaped structure. So antibody is having a Y-shaped structure. Okay. So in this Y shape, these two light pink color chain is known as light chain, and this blue one is known as heavy chain. Okay. So two small chains, or we can say light chain. Antibody contains two small chain or light chain and two long chain or heavy chain. So we can use a term too heavy and too light, H2L2. Okay, too heavy and too light, H2L2. Okay, and what is the shape? Y-shape structure. Okay, and this heavy chain and light chain contain various region. This light chain again contains a constant region, this darker one, and the lighter one is having variable sequences, so variable region. Similarly, this part contain constant region and this one is the variable region. And this is the top. These two act as a antigen binding site. Antigen binding site where any pathogen or antigen can bind. Okay. We can classify antibody into various types, mainly 5 types: IgA, IgM, IgE, IgG, and IgD. Okay, we can also use the term immunoglobulin. Immunoglobulin for antibody. So, either immunoglobulin or antibody, both are same. So we can use the term IgG as a symbolic name, IgM. That's why it is written over here Ig means immunoglobulin M or antibody M. Okay, immunoglobulin M, immunoglobulin A, immunoglobulin E, immunoglobulin G, or immunoglobulin D. Okay. Now, let's discuss antibody or immunoglobulin type and their function. Okay. So already I told you antibody and immunoglobulins are the same thing. These are having Y-shaped structure and these are recognized unique marker or antigen or pathogen. Unique antigen sequence, they very quickly recognize that. So first type of immunoglobulin is IgA or immunoglobulin A. These are present in the various secretions of our body, like mucus, like saliva, like tear, like colostrum. Okay, colostrum means milk secreted by the mother during their initial lactation days for the baby. Okay, so IgA is present in various type of secretions like mucus, like saliva, tear, and colostrum. This is having a dimer structure. Dimer. Okay. Second type of immunoglobulin is IgD. It acts as a B cell receptor. There are two types of lymphocytes. In last lecture, we have discussed T lymphocyte and B lymphocytes. Okay. So, IgD acts as a B cell receptor and it stimulates the release of IgM, immunoglobulin M. Okay. What kind of a structure it is having? It is a monomer form and Y-shaped structure. So, this is a monomer form. This is present as a monomer. Okay. Third type is IgE. E for IgE, E for allergy over here. Okay. So this mainly react or mainly involve in various allergic reactions. Okay. This type of antibody IgE bind to mast cell, bind to basophils and show different kind of allergic reaction and anti-parasitic activity. Okay. So these are also present as a monomer and Y-shaped structure. Structure is same. They may be present as a monomer, dimer or pentamer. So dimer, there are only two antibody. IgA is mostly present as a dimer and IgM as a pentamer. As you can see over here, 1, 2, 1, 2, 3, 4 and 5. Okay. Now, so IgE mainly involve in binding with basophils and mast cell and involve in various allergic reactions, various anti-parasitic reactions. Now, next one is IgG. IgG binds to phagocytes. And this is the main blood antibody for secondary response. Means these are present in a large amount. Okay. And this is the only antibody which are able to cross placenta. Okay. So, last one is IgM. IgM fix complements. This is the main antibody of primary response. Okay. B cell receptor and immune system memory. Why this is the main antibody in primary response when a pathogen attack to a particular animal or a body? Because this one is present as a pentamer, right? So it is able to, single antibody is able to bind 5 antigens or 10 small antigen. So very quickly, as the concentration is low, but one antibody is able to bind, single antibody is able to bind with the 5 antigen molecule. That's why IgM act as a main antibody during primary response. But during secondary response, large amount of IgG is present in the blood, although these are present as a monomer form, but concentration is very high. So they IgG act as a main antibody during secondary response. So concentration is high, but concentration is high, but structure is like this. These are only present as a monomer. So they are only able to bind only with a single antigen, or we can say 2 small antigen maximum. While here IgM is able to bind single antibody with the 5 big antigen or 10 small antigen. Clear? So these are present in the secretions. IgA, again I'll repeat once again. IgD mainly stimulate IgM, IgE for allergy, IgG for secondary response, IgM for primary response. This is the function of various immunoglobulins. Clear? Now, because these antibodies or immunoglobulins, these are found in blood, that's why we can use the term humoral response for the same, because these are present in the blood. Okay, during primary response or during secondary response, which is a type of adaptive immune response, we already discussed this, that adaptive immune response is classified into two different categories. One in which direct cell is involved, like T lymphocytes, and another one in which direct antibodies participate in the defense mechanism, that is humoral response. Okay, so this is a type of humoral response. During humoral response, which is mainly generated by B lymphocytes, is a type of adaptive immunity. Okay, and this humoral response is one of the two type of acquired immunity, that is antibody mediated. This already we have discussed in last lecture, right? And the second type is known as cell mediated immune response, or CMI, right? Very often when some human organ like heart, like eye, liver, kidney fail to function satisfactorily, that time what doctors suggest? Doctors suggest that transplantation, we have to transplant that particular organ. Kidney transplantation, liver transplantation, you must heard these terms. When their particular organ of an individual patient is not work properly. Okay, and in that case, relatives of that patient very quickly start searching for a suitable donor. Why suitable donor? Why they can't, doctors can't able to transfer anybody's organ to the patient? Because the body, our body is able to recognize, our body is having that memory that can recognize this particular organ is mine or someone else. Okay, and that's why the graft very frequently is rejected by the patient's body. So before transplantation, graft would be rejected by the patient's body. So before transplantation, doctors, before transplantation, what is the main test? Doctors did tissue matching, blood group matching, okay, before undertaking any graft or transplantation. And after that also, patient's body take immunosuppressant throughout life. Okay. Why? Because our body, as I told you, our body is able to differentiate self and non-self. And the cell-mediated immune response, CMI, is responsible for the graft rejection. Which response? Cell-mediated response. Clear? Now next topic is vaccination and immunization. So what is the principle of vaccination? The principle of immunization or vaccination is based on the property of memory of our immune system. We have discussed so many times this, that our immune system memorize that particular antigen very quickly, and after memorizing, for a secondary response, our immune system is get ready with the antibody reaction for that particular pathogen or antigen. So similarly, our body show a defense mechanism based on the memory of our immune system, as I told you. So in vaccination, a preparation of antigenic protein of pathogen or inactivated pathogen or weak pathogen are introduced into the body. In vaccination, antigenic protein, any antigenic protein from a pathogen or whole pathogen in inactive form or in a weak form are introduced into the body. Okay, the antibody produced in the body against these antigen would neutralize the pathogenic agent during actual infection. Okay, so this is the principle of vaccination. Vaccines also generate memory B cells and T cells. Memory B cells and memory C cells. T cells that recognize the pathogen quickly on subsequent exposure and overwhelm the invader with a massive production of antibody. So if a person is infected with some deadly microbe to which quick immune response is required, okay, some deadly microbe for which some quick immune response is required, as in the case of tetanus, we need to, in that particular case, we need to directly inject antibody inside the body. We cannot wait for the body to produce antibody via active immunity process, because that disease is very harmful. Okay, so in that case, we directly inject antibody or antitoxin inside the body. Similarly, in the case of snake bite, injection with the antitoxin or antivenom is required immediately. Okay, and this type of immunity in which direct incorporation of antibody occur, this type of immunization is called passive immunity. Okay, so recombinant DNA technology, RDT, has allowed the production of antigenic polypeptides of pathogen in bacteria or yeast, means vaccine production. Okay, with the help of RDT. RDT or recombinant DNA technology has allowed the production of antigenic polypeptide of pathogen in bacteria or yeast. Okay, so these vaccines production using this approach allow large scale production, hence greater availability for immunization for various diseases like hepatitis B also. And this hepatitis B vaccine produced from the yeast. So in this way also, recombinant DNA technology or biotechnology tools help us in medical field. Okay, now, so this is all about the today's video lecture. I hope most of the content has been cleared now. Still you are having any doubt, any confusion related to today's topic, you can write down to me comment box. We will discuss it later on. Till then, take care, bye bye, and keep learning. Namaskar.